Assalamu alaikum everyone. I would like to share my work about folate supplementations and hydrocephalus. The work has been conducted in University of Manchester. However, I am presenting through the platform of NUST. So what is cerebrospinal fluid? Let's start from the very beginning. Cerebrospinal fluid is basically just like water, and it's produced by brain at a rate of 500 milliliter per day. However, interestingly, the capacity of brain to hold CSF at the given time is only up to 150 milliliter, which highlights the importance of the regulation of CSF production and drainage. Um, that has to be drained every 7.5 hours. If we look at the production of CSF and the flow of the CSF, it is produced by the choroid plexus in lateral and third ventricle. From there, it's drained into the subarachnoid space and it leaves the brain either right from the top of the head, or it can pass down to the fourth ventricle where it leaves the brain through the hindbrain. Now, basically, hydrocephalus is just an abnormal accumulation of CSF within the ventricles. It can either be congenital or acquired. Congenital hydrocephalus is when the hydrocephalus is present from the birth, or acquired when it is attained because of any trauma or any infection. So the cause of hydrocephalus has rarely been the overproduction of CSF, but mainly the most common reason of hydrocephalus is subnormal CSF resorption in the body. The basic treatment of hydrocephalus is just the removal of extra CSF from the body by the placement of shunts, and these shunts, they will drain the CSF from the ventricles down to the peritoneal cavity where it will be absorbed and flushed out of the body. However, this treatment of shunt placement is quite complicated. It has life-threatening outcomes because of the complicated surgery and also because of the post-treatment infections that the patient can acquire. Now, if you look at the diagram here, this is a diagram of normal brain with no hydrocephalus, where the ventricular sizes are normal and the cortical thickness is normal. However, when the child achieves hydrocephalus, there is a lot more production of CSF in the ventricles, and the ventricle CSF will press the cortex down to the skull and resulting into the reduced cortical thickness. This is seen in clinical situation just like with the normal ventricular thickness and controlled amount of CSF present around the brain. However, in a hydrocephalic brain, we have really enlarged ventricles and accumulation of high amounts of CSF around the brain. This is a gross appearance of hydrocephalus, which is indicated by enlarged head size and abnormal facial structure. Now, why hydrocephalus is important? Why is it important that we study about hydrocephalus and understand it and find out different treatment or prevention approaches for that? So this bar graph indicates the prevalence of only pediatric hydrocephalus. The events are recorded as per 100,000 births, and in Asia it's around 67 births, live birth. And when I say live birth, it is important to record that. It means that there are incidents of preterm abortion. So what this chart indicates that it's a most common birth defect, but this is only recorded from the live births. And when the patient finds out that the baby to be born has a susceptibility to have hydrocephalus or the baby has hydrocephalus, so the parents mostly will go for the preterm abortion. Now, the global prevalence has been recorded as 88 incidents out of 100,000 live birth, and in Pakistan the frequency is 27. Nevertheless, the patients who are still alive, who they have the decreased intellectual capability from 62 to 30%. And it is important to understand that more than 50% hydrocephalic cases that are reported are congenital or pediatric hydrocephalus. which has been linked to the malnutrition in children as well as in the childbearing age. Now, we have developed a model of Hydrocephalus texas rat that recapitulate the Hydrocephalus as observed in clinic. So basically, this rat, the origin of this rat is a Sprague-Dawley rat, and there was a spontaneous mutation which was homozygous carrier of an autosomal recessive hydrocephalic gene. And the progeny of this animal model is divided into two further groups. One is named as unaffected hydrocephalus, and the second is named as affected hydrocephalus. And now the basis of their categorization is just how severe the condition is. If you see this figure here, that is a brain section of normal rat, Sprague-Dawley rat, with controlled with minimal size ventricles. However, if you see this figure part B, this is a section of an unaffected hydrocephalic rat brain with slightly enlarged ventricles. However, they are not as enlarged as to be categorized as affected hydrocephalic rats. So this brain section here indicate really enlarged ventricles, and then the gross appearance, the head appeared to be dome shaped, which indicates the presence of extra cerebrospinal fluid. Now, this morphology resembles quite much to the clinical representation of hydrocephalus. Now, a little bit about the folate cycle. Folate is basically an umbrella term for different forms of folate, including folic acid, which is just a synthetic form of folate, is a man-made folate, never found as such in the body. However, dihydrofolate, tetrahydrofolate, and then the further forms of folate, they are the biologically active forms of folate. And among them, the form of folate that we take from the food is tetrahydrofolate. So any kind of folate which has this THF group with it is biologically active. However, this figure here, drawn by the help of the data we have concluded so far, indicates that the conversion or interconversion of these subforms of biological active form of folate needs to be presence of certain enzyme, and one of them is FTH. Now, FTH and MTHFT1 have to be there so that this tetrahydrofolate can be converted into different forms of biological active form, and then they can represent as one carbon compound for methylation, for neurotransmitter synthesis, and for normal regulation of the brain functions. Now, important here is to memorize that FTH is then an enzyme that carries out the interconversion of different forms of folates. Interestingly, in this study, in the animal model of hydrocephalus, we found out that hydrocephalus is associated with a fault in FTH enzyme. So there are two publications that conveys this message to us. However, only selective data is shown here. So the figure here indicates that FTH has a capacity to bind with folates, which is further shown with this immunohistochemical data. This is the histogram. This is the histological analysis of cortex and brain cortex. What we can see here that FTH positive vesicles, they are uh, yeah, they are breaking off. They are breaking off the cortex, and then they bind with folate here, and they carry the folate. What we can see in this figure here, FTH has a capacity to bind in with the folates, not only in the cortical region but in the other parts of brain as well. And FTH has a capacity to bind with FR alpha, which is folate receptor alpha, that is usually considered as the folate transporter. And then here we see that FTH can bind with FR alpha as well. So what this set of data indicates is FTH positive vesicles will break off the cortical region, they will go into the CSF, and in the CSF they will bind with FR alpha and folate, and they will transport the folate across brain. So we find out a unique folate handling system in the CSF of these animals. Um, so now, interestingly, what we observe here that hydrocephalus is also associated with a general fault in FDH enzymes, which means if you consider this upper panel that represents the um, subcellular lysates of hepatic tissue, and this lower panel indicates the subcellular lysate from the cortical tissue. So what we see that as compared to Sprague-Dawley rat, in this colony of Hydrocephalus, we see a decrease in hepatic nuclear FDH. Similarly, we have a decrease in cortical nuclear FDH. And when there is a decrease of nuclear FDH, there is an increase of nuclear folates. That's what we see here, which indicates that the level of FDH are associated uh, low levels of FDH are associated with high level of folic acid. Now, if we see here, that's a dot product analysis for 5-methylcytosine. 5-methylcytosine is a marker for methylation. And what we observe that as compared to Sprague-Dawley animals, these unaffected and particularly affected animals have a really decreased methylation status, not only in liver, but in the brain as well. So we can link the expression of FDH with the expression of 5-methylcytosine. So this data indicates that we do need to have nuclear FDH for the proper methylation. So what we conclude from there is that nuclear FDH has to be there for the proper transfer and utilization of nuclear folate so that the cell can perform the methylation. Now, this set of data, and I do not know why this figure is not visible here, um, although it is visible when I not on the slideshow, and I apologize for that. But what we have done, we have some fetuses. So as soon as they're born from P0, they are treated with this certain supplements of folate supplementations, and from P0 to P10, like P indicates postnatal. So postnatal day 0 to postnatal day 10, they are treated with the folate supplementations. And what we see that in the brain of these fetuses, we can have the nuclear FDH present. So if we give them folates, then the nuclear FDH expression is improved. And there is a furthermore data that indicates that the cortical thickness is also improved, but that data is not published yet, so it's not being included here. So apart from treating the offsprings, the fetuses, we have an independent experiment where we treat the mothers, the female rats, before mating. So we are loading folates in the childbearing female rat before mating. And what do we see? If we have a combination of different kind of folates, if you go back to the folate site now, so we have a group that was the mothers were treated with saline, and then we have a group where the mothers were treated with folic acid. Now, folic acid is very well associated with the neurological disorders, and particularly the congenital disorders. That the using folic acid during the childbear age or before pregnancy can decrease the incidence of neurological disorders. So, keeping that in mind, we have folic acid itself as a treatment option, and then we have different forms of biologically active folates. So, this is folinic acid, and this is tetrahydrofolate. That is a mix one, which is a mix of folinic acid and tetrahydrofolate at a certain concentration, and the mix 2 is again a mix of folinic acid and tetrahydrofolate as a different concentration. So what this piece of data indicates that if the mother is given with folic acid, is given the supplementations of folic acid and then put into mating, then the chances of hydrocephalic babies are around 50%, right? It's increased 50%. as compared to the mothers who were just injected saline. Now, if we have given folinic acid, the chances of having hydrocephalic babies reduce as compared to saline and folic acid treated mothers. Tetrahydrofolate is also improved the reducing the occurrence of hydrocephalus, but the mixed one has significantly reduced the occurrence of hydrocephalus, and so has mixed 2 with a different percentage. So this data is a very important piece of information, in fact, and what we can conclude here that folic acid that might have previously been associated with neurological disorders, particularly the developmental disorders, that it can improve the developmental disorders. To highlight one of them is spina bifida. The folic acid could be a detrimental option for those who are susceptible to have hydrocephalus. However, using these biological active forms, and particularly this particular mix that we have used here, has significantly reduced the affected fetuses that are produced in the colony. So this piece of data is certainly clinically really important, that it is not about folic acid, it is about the right kind of folates that needs to be used to avoid or to treat the neurological disorders. So what we have further found in this colony is that not only the mothers that might be affected because of low levels of folates, it is also the parents who need some extra supplementations. What we find out here, the parental folate-related methylation fault is associated with hydrocephalus. So this study indicates that as compared to the normal animals, we see, as compared to the normal animals, this A and C here, we see a distorted testicular morphology in the hydrocephalic rats. And these rats are unaffected hydrocephalic rats, because the affected hydrocephalic rats, we cannot use them for mating and they do not actually survive for that long. So basically what we see, these are unaffected hydrocephalic rat, but they do have testicular morphology, distorted testicular morphology. And when we see, this is the tunnel staining, and when we see the difference between the intact and disintegrated DNA, what we find out that in Sprague-Dawley animals, we see a lot of cell death, a lot of DNA damage in this unaffected hydrocephalic rats testes. So what we further find in these rats is a differential expression of FDH, folate receptor alpha, folate itself, and what we find out that although they might be more or less, like FDH is increased in untreated, unaffected animals, then we have a group that where the males were treated with that particular mix before mating, and what we see that FDH expression is regulated. So what we see here, it is in the nucleus, but then in unaffected, it's not in the nucleus, exactly what we observed in the fetuses before. And when we treat these males with folic acid, with that particular folate, what we see that the nuclear expression of FDH is further restored. Similarly, as compared to Sprague-Dawley, there is a lot more folate receptor alpha, which is just present there, but that condition is reversible with the treatment. These pictures indicate where the folate is present. Here the folate is present in the like quite scattered, but here the folate is in the nucleus, which indicates because this folate receptor alpha is also present in the nucleus, and this is taking folate to the nucleus. And what we further see that when we give the treatment to the males here, then the expression of folate is further scattered. So there might not be actually any difference of the folate levels, but the difference is where the cellular localization is. Now, what else we see here, that if these are the Sprague-Dawley rat testes, we see that the sperm head and the sperm itself, they have 5-methylcytosine, a lot of 5-methylcytosine in all the sperms seen here. And the SD rat testes, they do not have much 5-hydroxymethylcytosine present in the sperm, which indicates that there is a lot of methylation going on, but there is no demethylation going on in the Sprague-Dawley rat testis, which is considered as normal. What we see in the untreated, unaffected hydrocephalic rat testis, that there is such a reduced amount of, such a reduced expression of 5-methylcytosine, which is indicated here, like the sperms are not being methylated and the methylation is only happening in this primary spermatozoa. What we see, there is a lot of demethylation going on. So if you see these cells, the primary spermatozoa and the sperms, they are going under a lot of demethylation as compared to the Sprague-Dawley animals. Now, how the treatment has affected? So in the rat testes, which went under folate treatment, we see that the levels of methylation has improved. So we do see the sperms that have 5-methylcytosine expression, indicating a restore of methylation. And we see the reduction in 5-hydroxymethylcytosine, which indicates a reduction in the demethylation. So this is dot plot analysis indicating reduced methylation in untreated, unaffected hydrocephalus, and a lot of improvement in methylation in treated hydrocephalic animals. However, as compared to Sprague-Dawley, we have a lot of 5-hydroxymethyl expression, and which is reduced because of the treatment. So this piece of data indicates that the susceptibility of having less methylation and more demethylation could possibly be related to the developmental defects, which is partially reversible with the folate treatment. Now, this data indicates that we do not only need to give the supplementation to the childbearing female, but the sexually active male should also get this particular kind of folate supplementation to improve the sperm quality and to improve the consequences of the treatment. So we have successfully seen here that the folate supplementation, and that to the right form of folate supplementation, is very important in the sexually active animals. Now we want to find out if these results are in line with what we observe in human samples. So we have, excuse me, so we have these normal human samples, CSF, and these human samples are from hydrocephalic humans, and these CSF samples are from embryonic day 20 rat fetuses. So this lane represents control, and these two lane represents affected. So in the normal human, there is abundant FDH present. However, in hydrocephalic human, the FDH is reduced. Similarly, it is aligned with the data that we have observed in E20 rat fetus, that the control has a lot of FDH, but the affected fetuses has reduced FDH expression. So this gives us an understanding that there is a common fault present in this HTX colony of rats that we have as an experimental model, and the biological samples that we get from the human patients. In fact, the IPA analysis has identified folate unique pathways. So these pathways are unique in abnormal CSF. So they make folate inaccessible. So the folate might be there, but it is not accessible to the brain. What does that mean? Now, that means let's say if we are already taking, if we are not taking folic acid, so we understand that folate is not present, folic acid is not present, so it's not being taken up by the brain. However, if we put patients on the folic acid supplementations, and even then the pathway problem does not make this folate available to the brain cells. So um, we have to bypass these pathways. And how can we bypass these pathways? We have to use the active form of folate, which we have used for this experimentation. So with this hypothesis in mind, that there is a common fault in human CSF as compared to rat CSF of this hydrocephalic patients. We have a hypothesis that if we give these patients the same the same particular mix, there should be a reduction in the hydrocephalic incidences. Um, so our research hasn't not only used these hydrocephalic samples from the patients, but we also have worked on Alzheimer's disease. So these two papers indicate they have found out the folate-related pathway faults in Alzheimer patients, which further confirms our understanding, supports our understanding, and support the hypothesis. And we are working towards more and more human types of neurological disorders. To name a few is traumatic brain injury, non-pressure hydrocephalus, normal pressure hydrocephalus, um, Alzheimer's, dementia, and multiple sclerosis. And we have found a common folate fault. Now, what we conclude from this set of data is that hydrocephalus is a result of a treatable folate deficiency. So basically, this study also highlights that not only the mothers, but also the fathers need to be treated with that particular form of folate uh, to reduce the chances of hydrocephalus, and also we can improve um, the the brain structure and cognitive ability um, by giving the right type of folate to the fetuses affected. A fetus is born as hydrocephalic. The future plans are that we are still working on a lot more different samples from different neurological conditions, and not only on CSF but also in the brain. And the IP has already been registered internationally for this mix. Sorry. And the clinical trials are being planned in collaboration with Qatar Sidra Hospital, University of Manchester. So we are working towards that. And that is all about my presentation. That is all about the talk. The floor is open for discussion. And thank you very much.